Welcome, everybody. My name is Cornelia Thoeni. I am a PhD resident in the Anatomical Pathology Residency program at the University of Toronto. Today, it is my pleasure to present my previous research work in liver pathology. What was my role in the project? I was principal investigator and corresponding author of my previous published work, expression analysis of ATP-binding cassette transporters ABCB11 and ABCB4 in primary sclerosing cholangitis and variety of pediatric and adult cholestatic and non-cholestatic liver diseases. This work was in collaboration with my previous supervisor, Professor Dr. Ernest Kutz, who was previously working as a GI and liver pathologist at the Hospital for Sick Children. What was the significance of the findings? We were interested to find novel diagnostic markers in a specific cholestatic liver disease, primary sclerosing cholangitis or PSC. PSC is an autoimmune liver disease characterized by acute and chronic inflammation of bile ducts. Here you see a bile duct with the bile duct epithelium surrounded by numerous inflammatory cells, and the bile duct wall has extensive fibrosis. This is called onion skin fibrosis, specifically seen in PSC. Furthermore, bile ducts are occluded with bile pigment. This is called cholestasis. Bile can also accumulating in hepatocytes. Then this is called intrahepatic cholestasis. As bile contains toxic products, this leads to severe liver damage and cirrhosis, as you can see here. Which methods did we use for our research project? We were interested which ATP-binding cassette transporters are differently expressed in PSC hepatocytes. ATP-binding cassette transporters are important to transport bile contents from the hepatocytes into the bile canaliculi. Therefore, we analyzed a cohort of 43 patients at my previous working place in Heidelberg, Germany. We had 15 PSC patients and 28 non-PSC patients. We analyzed their histology and we extracted individual hepatocytes with laser capture microscopy and extracted RNA. Finally, we analyzed gene expression analysis with nanostring technology and in parallel protein expression analysis with immunohistochemistry. Here are our results. We demonstrated that ABCB11 and ABCB4 were significantly upregulated in PSC patients. Here is the immunohistochemical picture. ABCB11 and ABCB4 are stained in brown. The nuclei are stained in blue. Here you see ABCB11 is significantly accumulating and expressed at the bile canalicular membrane in hepatocytes of PSC patients. The same is seen for ABCB4, strongly expressed and accumulated at the bile canalicular membrane of PSC patients. In summary, we were able to show that ABCB4 and ABCB11 were significantly upregulated in PSC patients. What was my favorite and most interesting part of the project? First of all, I learned on histology how to grade cholestatic liver disease. Therefore, I got familiar with the Nakanuma score and learned how to stage fibrosis. Here's an example for cirrhosis with regenerative nodules, how to assess bile ducts, inflammation, and also regenerative changes, as you can see here, extensive fibrosis, the onion skin fibrosis, and how to grade cholestasis with the Shikata's orsein staining. This is a special stain highlighting copper-binding protein in hepatocytes. Copper-binding protein is upregulated in cholestatic hepatocytes, and here labeled as purple-pink granules. Next, I was excited to learn how to use nanostring technology for gene expression analysis. It is very simple. You have two probes, your universal capture probe for your gene of interest, and then the reporter tag, a fluorescent label probe. This is important for quantification of your gene of interest. And it's very simple to analyze with the NSolver program. And finally, I used my previously published work to apply for further financial support. I applied for the LNP Innovation Competition Award 2021. This is a specific award from the University Health Network, the pathology department, and applied together with my wonderful mentor, Dr. Sandra Fisher. She is an excellent liver consultant pathologist at Toronto General Hospital, and our application was successful. This is our contact information in terms you have any questions about our liver project. And I hope you liked my video and the exciting world of liver pathology. And finally, I want to thank you for your attention.